예, 안녕하세요. 이번 웨비나에 귀중한 시간 내어주셔서 참석해 주셔서 감사하다는 말씀 드리고 싶습니다. 자, 그럼 웨비나 시작하겠습니다. 예. Hello, everyone. My name is In-Seok Choi. I'm a postdoc at UC Berkeley in the Department of Bioengineering in Professor Seung Lee's laboratory. In this presentation, I'll introduce our work, bio-inspired design of functional materials using bacteriophage. So let's start. So in nature, we can find a lot of smart biomaterials. Some of them are evolutionary. So like, for example, bacteriophage can modify their structure and attack different and new bacteria. So they are excellent tool to prevent the bacterial infections and treat. Some of them are structural. For example, cellulose nanocrystals, they are produced as a crystal and then assembled in a certain manner that it can induce these optical responses in a much bigger scale. So as an engineer and scientist, our role is to find the pathway to use these smart biomaterials for functional material applications. We are using virus. So maybe some of you might think the virus is such a dangerous and hazardous material as we experience the COVID-19. But there are many safe and good viruses that we can utilize for advancing the functional materials. So this is the mechanism of utilizing the virus. So virus has coat proteins on the surface. So if we genetically engineer the DNA of the virus, then we can express the desired peptide sequences on the protein. And once it has this specific peptide on the surface, it can induce the selective binding. So here, I'll introduce how we can utilize the selective binding for chemical separation first. And second, how we can utilize this supramolecular assembly of the virus for tissue regenerations. So I have two different sections in this presentation, and I'll first introduce this virus for rare earth element separations. Okay, some of you might not be very familiar with what is the rare earth element. So rare earth element is very important element that we need to advance our modern technology and have renewable energy devices. So maybe the most common rare earth element that we can think of is the ND magnet. The ND magnet has a strong magnetic property that can be used for battery and electrical vehicles. And some other elements can be used for the display as it generates a fluorescence effects. It was chosen as the 70 most important chemicals that can change the world in 2016, this Nature paper. And here, these rare earth elements are positioned in the lower part of the periodic table. So before I start this project, I didn't even know what is the lanthanides before. So it is very common that many material scientists do not know what is really the rare earth element. But they are here, the lanthanide groups and actinide groups. And they are mainly used for catalysts, lasers for the defense purpose, MRI for biomedical, and also display and, like I said, magnet productions. So we need this rare earth element to advance our modern technology. But there's a big issue in extracting the rare earth element from the earth. So for example, here, US led the rare earth element market since 1960, and they stopped, right, at 1995-ish. The reason is because of the environmental issue. There was a huge leakage of the chemicals in the California site. The US government shut down a lot of mining sites. And this environmental issue is repeatedly occurring in many other regions in the world. Because this rare earth extraction requires huge amounts of hazardous extractants, acidic and organic solvent, they need to have this chemicals to extract the metal ions from the natural ores. And also it generates a lot of radioactive wastes. So there's a big environmental issues. So we need rare earth element, but because of the issue, the supply chain of rare earth is not very stable in the world. So then as a biomaterial scientist, we can ask this question. Can we utilize biomaterials to develop environmentally friendly RE separation process? With this question, we start this project, and this is the key idea of using the virus for the rare earth element separation. Like I explained, as we design this RNA, we can express the peptides on the surface. And if this peptide can selectively bind this rare earth element, simply REs over non-REs, basically we can separate REs, right? But the big challenges remain as there's a notable chemical similarity between REs, and having the high selectivity to separate different REs from each other is very important, and it is the most challenging part. As you can see here, the REs are mainly separated as two groups, the light REs and heavy REs. And how we can separate this heavy RE and light RE, that is also a challenging part. Then how can we get this information? What protein we need to insert to the virus? Then it can express this RE selective binding motif. In 2005, I would say the nature gave us a present. This Italian scientist went to the Berkeley mudpot and collected many different bacteria. And he accidentally found several bacteria can survive only with lanthanides. It was not on purpose, but accidentally found a certain bacteria, for example, methylotropic bacteria, can survive only with the lanthanum. It is very interesting because rare earth elements are much, much smaller on Earth compared to other metal ions, but some bacteria can survive only with these lanthanides, right? And biochemists like Professor Kotrovo's group, they jumped into this field and found this unique protein code that can bind the rare earth element inside the bacteria. This DPKD-GTID-LKE, this is the amino acid sequence that can selectively bind rare earth element versus over other metal ions. So we, as a virus group, we genetically inserted this unique sequence into the virus, and we could create this lanthanide binding virus. And some of you might ask, why should we use the virus? What is the advantage? So here's the answer. This virus, once it has this specific binding motif, it has this around 3,000 to 4,000 coat proteins on the surface. So with this high selectivity, it also has this large capacity to bind this rare earth element on the surface. Okay, and second, this is a virus, so it inherently have this advantage of producing many viruses through the infection of the E. coli. So within one day, we can make more than billions of the viruses by just mixing virus and E. coli in an incubator. So it's very easy to produce large quantity of the virus. Okay. So this is the reason that we investigate further using this virus for the RE separation. And we first demonstrated the selective binding, the heavy RE versus the light REs. Through the circular dichroism analysis, we found this engineered virus has originally alpha helical structure on the major coat protein. And once these lanthanide ions are introduced in the system, it can change the structure to the random or beta form. So when this heavy RE for example, terbium is introduced as a 7.5 equivalent to the code protein, it can change the structure, and we could see the structure change. And different from this heavy RE, the light RE, we need a much, much higher amount of the ion to induce the structure change. So that means this heavy RE can, excuse me, the heavy RE can generate stronger and much preferentially generating the structural change of the code proteins of the virus compared to the light REs. Okay. We also performed many different lanthanide ions on the surface of the virus, and you could see the alpha helical content significantly drops at much lower concentration range of the heavy REs compared to the light REs. And also, through this dynamic light scattering, we could see how much aggregation is occurring by introducing our lanthanide ions to the virus. And you can see the huge aggregation is occurring with the heavy REs at much lower concentration of the light REs. So our result shows our virus has the preferential binding and interaction to the heavy RE than light REs. As next, we tested how much the lanthanide ions can bind to the surface of the virus. You can see here the adsorption characterization. The heavy REs were bound to the virus more than light REs. We tested this by confining our virus inside the dialysis tube and analyze how much binding is occurring inside the dialysis tube by comparing heavy RE, light RE, and non-REs. And our result shows non-REs were not bound to the virus, but heavy REs were bound to much, much more than non-REs and light REs. Also, like I said, our virus has the advantage of high capacity of the binding. It has around 3,000 proteins on the surface. So we could see around 3,600 lanthanide ions could bind to the virus. So it has such a high capability of binding a large amount of the REs from the mixture. And we could also control the binding and releasing of the lanthanide ions through the pH modulation. Owing to this charge change of the amino acid, like D and K, by decreasing the pH, we can control the amount of the charge on the surface. At pH 5, the D and K, the K is the positive, so D is the major part binding to the light REs, light lanthanides. So they have negatively charged at high pH, but they lose their charge at low pH. So by controlling the pH, we could adjust the adsorption amount on the surface of the virus. Okay, at pH 5, this large amount of the REs were bound. And by decreasing the pH, the bound REs were removed from the surface. So using this pH modulation effect, we could demonstrate the adsorption and desorption in multiple cycles using our newly created virus. At pH 5, like I said, because of the negative charge and the inserted the peptide, the heavy REs were bound more than the light REs inside the dialysis tube. And as we decrease the pH to 2, there is a releasing of the REs from the virus. Then we can separate basically from the non-REs too. So you can see here over the 5 different cycles, there is no decrease of the binding efficiency. And also the heavy REs groups, the red, are bound more than the light REs. So it shows this excellent recyclability for large-scale separations of the REs. And as last, we demonstrated our virus-based RE extraction from the naturally made this allanite ore. This allanite ore has different lanthanides and non-Rs. As you can see, the leachate solution of this allanite has much higher amount of non-Rs versus Rs. And as we induce this adsorption and desorption cycle through the pH modulation, we could separate selectively the Rs from non-Rs and also more heavy Rs than light Rs. So this is a summary of the first part. We could genetically engineer the lanthanide binding virus, which exhibits stronger interactions with heavy Rs than light Rs. And the virus demonstrates preferential and high capacity binding of Rs, and it highlights the potential for the development of this virus-based RE separation technique. So we found this bio probe for the RE separation. As next, we are working on now scaling up. So utilize our virus for ion exchange process and also phase separation process to induce a larger amount of the RE separation in a bigger scale. So we are also very excited on this project because we can expand this chemical separation using virus for different target materials. For example, microplastic separation and the toxic metal separation for water purification. That is the next thing that we can work on. And also protein purification and the virus detection. So we basically use this virus as an adsorbent and also detecting probe for different materials. So this is the exciting future of our work. Okay, let's take a 20-second break before going to the next section. Okay, so let's start for the second part. In this part, I will introduce the virus for structural tissue engineering materials. The screen is frozen here. Okay. Yeah, here. So in nature, many biomaterials are forming this hierarchical structure. And this hierarchical structure is very important as this biomaterial induce functions in multiscales. And this is an example of collagen. So collagen, as you can see here, with hydroxyapatite, they form the bond. Individual components have a lower functionality in the mechanical strength, but as they combine and make a composite and assemble, they can make such a strong bond structure. So our bond has much higher mechanical toughness than even the mineral itself. How this is happening? The collagen is forming this nano building block, and they are assembled in a triple helical manner. And also, they have strong binding to the hydroxyapatite. So first, we need to have this binding, and second, as the building block, and they are forming this composite and assemble into the bigger structure. So this building block binding, followed by this self-assembly, are really the key to form this hierarchical structure and inducing the function in multi-scales. Let's discuss more about the collagen. The collagen is the most abundant protein in the human body, and they are forming different structures in different parts of the body. For example, in cornea, they are forming this orthogonal structure by themselves. And in cornea, they are transparent and they change the light refractions on the surface. So they are functioning as a lens, right? And human tooth, they are forming this fibrillar structure and making this highly packed the fibrils in the dentin. And they are making high tough the tooth in the human body. Okay, so like these examples, the proteins are really the key important material in the human body and for the tissue engineering. So many researchers have investigated the collagen alternative material to make the tissue engineering materials. Because collagen, the native collagen have many issues in the human body. Different types are mixed and they can generate a different side effects. So many collagen mimetic peptides have been reported. And you can see here, for example, PHG is having the same similar sequence to collagen, have this different molecular structures. So there have been many success in the research developing these collagen mimetic peptides and demonstrated functions in the molecular scale. But the main challenge is remained. It is still difficult to achieve the hierarchical structure, which is the key for the bigger scale functions. So then we start this project with this question. How can we construct collagen-like structures for multi-scale functions? Like I said, in the nature, the building block is the first part, and second is a strong binding is the second important parameter. And the third, the assembly, is the key for the multi-scale functions. So here I want to introduce our bacteriophage as an excellent biomimetic building block for the tissue engineering materials. The bacteriophage has the DNA inside, and it has this specific size of the whole material. You can see it's very uniform, and these coat proteins are expressed on the surface, as they are called the capsid proteins. And these are the safe virus, so they are already commercially used for the phage display and for making the antigens and antibodies in pharmaceutical companies. So they are very safe. And because it's a virus, it has this strong advantage for the manufacturing. Like I mentioned in the first section, this one phage can replicate over billions of times using the E. coli as a host in one day. And also a material scientist view, this is an excellent material for the tissue engineering because the surface interactions can be totally changed by genetically inserting certain part, then we can express our desired peptides on the surface. It has the major coat proteins, around 3,000 copies on the surface of the virus, and each protein, the peptides can be modified by inserting the certain gene. So then eventually we can have all the proteins having the same sequence of the proteins. Okay. And also we can control the length from the 50 nanometer to 2.5 micrometer. We can change and control the length of the virus. So it is an excellent building block, especially for the making the assembly in a bigger structure. Okay, so this is our strategy to make the collagen-like virus. So we combined these two techniques. The first, phage display, and second, self-templating, to create collagen-like tissue engineering materials. Phage display is a well-known technique, simply called direct evolution. It uses the genetic diversity, and through the high throughput screening, we can select the best binding motif to the target materials. And the self-templating is a relatively new technique developed by my PI in our lab in UC Berkeley. It is called the depth trick to use the liquid crystalline behavior of the nano biomaterials. So in the process, we are pulling the substrate and in solution at the meniscus because of the evaporation and the position on the substrate. Under the high concentration, they are forming this liquid crystalline structure. So we can tune the optical property and also dimensions of the structures through this templating assembly. So here's the process of the phage display. So like I mentioned, the collagen is collagen is binding to the hydroxyapatite, the bone mineral, first, and then make a structure. So we first identified the best binding virus to the hydroxyapatite minerals through this phage display. So first step is to randomize the gene of the code protein and then make this library. This library now have millions and billions of different phage in one part, and then introducing this one part and with different phages onto this bone mineral, and screening, we can find the best binding phage to the hydroxyapatite mineral. And we performed this work and found this phage having the highest binding and named it collagen-like phage. This collagen-like phage have a very interesting feature. As you can see here, the sequence that we independently found through the phage display has the similarity to the naturally evolved collagen. So natural collagen has this PXY sequence. In our phage display result, this virus also has this PXY and PXAY sequence on the surface. And through this molecular dynamic simulation and also experimental work, we could identify these OH groups from the serine, repeating serine, could be a key to induce the strong binding to hydroxyapatite crystals, as it has very similar the length between two different OH groups and O in a crystal. It can make strong hydrogen bonding. This hydrogen bonding could play a key role to make this virus strongly binding to the hydroxyapatite minerals. Okay? So we found this excellent biomimetic building block, collagen-like phage. The next task is assembly to make collagen-like structure. So then how can we make a collagen-like structure? So here I want to show you what is the distinct feature of collagen assembly. Not all of them, but majority of the collagen form these fibrils in a micro scale and also the macro scale. So these individual triple helix collagens are assembled in a very specific manner called despacing, and then make this stepping structure that it has the overlap and gap regions alternatively with a very specific size. So as you look at this collagen structure, it has these periodic gaps, exactly the same distance, right? And it's called the 1/4 pitch because it's 1/4 of the full length of the triple helix collagen. And this periodic gap is important as it provides the nucleation site for the infiltration of the bone crystals. That means on these fibrils, the bone crystals can grow from these gap regions and make this composite along the fibrils. And as we tested the sample templating assembly of our phage with the two different lengths, one as a 240 nanometer and the second as 950 nanometer, we could see these beautiful collagen-like fibrils. So we are very happy to see this collagen-like structure because it has the three features. Directional order is possible, and spatial order with the 240 nanometer repeating gap is possible, and also these periodic gaps. And spatial order, periodic gaps, and directional order, those were achieved through the self-templating assembly of these two different length phages. And we could make very collagen-like structure using our virus. So this is the exact mechanism of making these collagen-like structures. Like I mentioned, there are two different lengths. One is long, and the second 950 nanometer. The second is short, 240 nanometer. And this long phage is assembled on the substrate. They are aligned along the pulling direction of the substrate because they have a higher shear rate, a shear stress on the phage particles. So they are aligned along the pulling direction. So that contributes to the fibril elongation. The short one, they have the spectral packing on the surface of the elongated fibrils. So they are making this synergetic effect and make these periodic gaps along the fibers. So then eventually, by the pulling, we can make these periodic gaps, overlapped and gap regions, spectic packing along the fibrils. Then the next question is, how can we use these collagen-like fibrils for the tissue engineering? So first thing that we tried was to see if this phage can nucleate the bone minerals. So we introduced calcium and phosphorus ions on this phage fibrillar matrix, and could see those ions are bound to the matrix and grow as the crystals. And through this EDX characterization, we could see it is really a crystal formation on the surface along the fibers. Not just inducing this nucleation, this collagen-like fibrils could work as a template to align the big osteoblast cells. As we grow the cells on this template, those cells could grow along the pulling direction of the fibers. So these cells could feel this physical cue from the alignment of the fibers, and they grow along the fibers, and we could make this directional order of the cells of the bone tissue, of the bone cells on this fibrillar template, and also make this directional order of the crystals along the fibers. Okay. As last, we could demonstrate the actual application of our collagen-like phages. So this is the case. As we grow older, as getting older and older, there is a demineralization occurring faster than the remineralization on our tooth. So enamel is full of the inorganics, but in dentin, there is a collagen in other organics, and also there is making composite with the hydroxyapatite crystals. And what happens is when the demineralization occurs faster, then it generates lots of holes. And because of the holes, it's called the tubules. These tubules, when you eat the food, the liquid go inside and generate the hypersensitivity. That's why we feel coldness when we eat apples. Okay, and it's becoming worse and worse as we get older and older. So to prevent this, we need to block it and then remineralize the surface. So we got this tooth as a disc from the dentist, and then deposited our collagen-like phage and make a structure so that by the structure, these holes could be really filled completely. And then we grow these hydroxyapatite minerals on the surface, so we could eventually make a high toughness bone structure using our phage and hydroxyapatite minerals nucleations. So we could successfully block these dentin tubules with our phage fibrils, and then prevented hypersensitivities. Okay. Okay, this is a summary of the second part. We developed this collagen-like building block using phage, and the phage display, we identified the best binding phage on the bone mineral. And after we induced the self-templing assembly to induce the bigger structural fibrillar formation and demonstrated crystals and cells growth along the fibrils. So we can now control the growth of the bones and growth of the cells directionally. And this is a very exciting future. So we demonstrated the bone and utilized our virus for the bone tissue engineering. But we know different parts of the body, the collagen is packing differently and function differently. For example, the collagen is working as an optical lens in eyes, and they have different packing, right? And we can make different packing by controlling the self-templating assembly parameters and use different the alignments, so that you can induce different functions on the body. So we will expand our tissue engineering material using the virus onto different cells and different tissues of the human body. And we can demonstrate more with our virus-based tissue engineering materials in the future. Okay, so these are the two references that you can take a look if you're interested in this work, and these are published last month. So please take a look, and also please cite these papers if your work are related to our phase-based tissue engineering and phase-based the virus phase-based rare earth element separation. And I want to thank you very much for all of you to listening to my long talk. And I also want to say thank you to my PI and all of my collaborators, and also my undergraduate team at the UC Berkeley. Thank you very much again. And if you have any questions, please feel free to ask me.